Morning, Monday, the 29th. And here we go on our nervous system. We finished the first three body systems that we're going to worry about. Now we're going to move on to the nervous system. Kind of like the reproductive system in the last trio, this is the big one. This is the one out of this next group of three that by far has the most material, has the most stuff. It is the most important one that you need to understand moving forward. And today, particularly, you can click through the slides. I will get a video up going over and presenting them as soon as I can. But predominantly, I want to go over the labs because the nervous system labs can be more confusing than some. There are directions. To understand them, you must read the directions. History states that ninth graders are not so good at reading the directions. So let's go over it so that I can help explain it. We're going to be starting with the first lab this week. Now, I am assigning these two labs just to get them done. Just whenever you get them done, fabulous. Just get them done. So the first one technically is going to be our nervous system lab. We are still in Unit 6 now, week 4. The other one is the brain side dominance lab. You can do them in any order. If you're here in person, then we're going to go over them in this order. But yeah, like the Purple Day folks will do the White Day folks will do the nervous system lab today. Purple Day folks will probably work on it tomorrow. We might bounce around a little bit. Yeah, it's going to be a little bit messy, but we'll get it done. So let's go over this lab. Now, the nervous system lab is going to have you doing 4 different parts. The first part will be measuring you and a partner's reaction time. And we're going to measure 2 different ones. From the shoulder to the arm, closing your hand like this to catch something, and the thumb and index finger where you just have to do this. Now, the basic idea is that you would get a meter stick, that one person would hold the meter stick up above. All my meter sticks disappeared. We'll use this bottle. All right, we would hold it up above. The other person would have their hands like this. And then at some point, you would let go, and your partner would try to catch it. And then the second one, you hold it up above. They have their hand ready. And at some point, you let go. And once again, they try to catch it. Then we measure the distance on a meter stick. That's nice and easy because it's marked for you. And then you record it and average it out. Now, if you are online, though, and you say, but Mr. Matthews, I have no meter stick. However, shall I do this? There's a solution to that. Back in Schoology here, I included this reaction time online thing. And let me go by Syntac there. All right, and it will take you to this website. And basically, you're going to do your trials right here. Read the directions, follow the directions. It will give you your reaction time for the 4 trials you're going to do. You don't need to do the 5th one. And it will average them for you. So all of that is done. Now, the one thing this does not do for you is this does not handle the difference between a hand motion and an arm motion. So you're going to have to run it twice. Once with your hand on the mouse, ready to click when you see the change. The other one, you're going to want to start with your hand about a foot above your mouse, and you need to bring it down and click. And there go the lights. So those 2 differences will account, hopefully, for the difference between shoulder and arm and finger and hand. So that will be part 1, measuring your reflexes. And you'll record them here. Then you're going to need a partners. Get somebody else. Even if you just know somebody else from class, you message them, what numbers did you get? And then share back and forth. Next, we're going to map nerve endings. Now, this one's going to be a little bit more difficult. You can do it solo. You can use anything that's kind of pointy in order to do it. I strongly suggest toothpicks. If you don't have toothpicks available, a pencil, or sorry, 2 pencils will work for it. In this case, because the toothpicks that I have are already parted out for class, I just have 2 sides of a paper clip. Now, what you're going to do is at 3 different points on the pad, which is right here, that is the touchy part of your finger, the back of your upper arm, back here with your tricep muscle, and the back of the neck. Now, all we want to do is we want to determine at what point do you only feel one poke. So you start with your points a pretty good distance apart, and you touch them down, and right now I can definitely feel 2 of them. So we move 1 a little bit closer, then we do it again. I still feel 2. So we move it a little bit closer again. I still feel 2. So we move it a little bit closer again. Now at this point, I only feel 1. So now I will take this and I will measure this distance right here at the end. And I will record that for our first value. So, like that might be 2 millimeters. And once again, you're going to do 4 trials and average them together. Then you're going to do the same thing on the back of the upper arm. And once again, you can do this to yourself. You don't need somebody else for it. You're going to want to not have a shirt in the way, and then poke. And if you feel 2, move them closer together, poke. And if you feel 2, move them closer. Whenever you feel only 1 point, that's when you stop and you measure it. And then again, on the back of the neck. So I felt 2. But like right there, I'm feeling 1. Look at the difference between the pad of the finger and the back of the neck. All right, so that is our nerve endings. And we're just checking how close, how many neurons do you have in a given area? Because as soon as you only detect 1 point, then you only have 1 neuron to communicate back to the brain saying that it's being triggered. So, for instance, we know that on the pad of your finger that we use to touch everything, we have a lot of neurons. Those 2 points could be very close together, and we could still tell that it's 2 separate points. But somewhere like the back of the neck, we don't really use the back of our neck to feel much. So we have many, many fewer neurons there. So the distance can be much greater. Text box. All right, number 3. This one, now we're going to check temperature. First, you're going to need some flat disc-like object. Pennies are suggested, but anything like that will work. We're going to be using paper clips here in class. And what you want to do is first, you want to take your hand, dunk it in water, and then you want to count how many can you pick up in 30 seconds. Record that in the wet hand count. Then you're going to take your hand. Now we need to freeze it. Now, we will be using in here an insulated tub of ice and water to really chill the hands. If you are home, you can get a little bit of ice. You can get some water. You can dunk your hand in it, whatever works for you. But what you want to do is you really want to get your hand really substantially cold. Now, safety warning. Do not submerge your hand into ice water for longer than 2 minutes. The lab only wants 1.5 minutes. Do not do it for longer than 2 minutes. We do not want any kind of nerve damage. We just want to freeze things bad enough that stuff doesn't move as well. And then once again, once your hand is super cold, Try to pick up those same objects for 30 seconds and see how many do you get. And then, of course, get your partner's information. Now, the last one is mapping the blind spot. Now, this one is a little bit more general. You'll notice there's nothing to write down here. Basically, you have a blind spot. There's an area in your eye where the optic nerve dives backward to your brain. The optic nerve can't see anything, which means that you've got this circle, roughly, that has no ability to communicate any sort of visual information, and it is there all the time, somewhere in your periphery, but your brain is constantly filling in that blind spot with whatever it thinks should be there. So what we want to do is you want to take your fingers generally like this and just stare forward, just like a dead stare, like you're so bored, you're in my class, you just can't handle it anymore. And what you want to do is start right towards the middle, and you want to kind of move them out and around the periphery. At some point, they'll get real fuzzy. For me, it's right here. And that's your blind spot, where you don't actually see them. You know they're there. Your brain will tell you they are there. But they aren't going to really, you won't be able to see them. And you just want to kind of keep in mind where that is. For me, once again, it's right about here. Then you want to kind of move it and see, like, at what point, don't move your eyes. Just kind of use your peripheral vision to tell when you can see them clearly again. And that's it. You're going to answer that. Then down here for the questions, there's one for each. First one, based on the little pokes. Second one, first one, reaction time. Second one, little pokes. Third one, cold hand. Fourth one, blind spot. And that's our lab. It's got a lot of stuff to do, but it's really not too hard. So I hope that clarifies it. We want to get this done before the end of the day on Thursday. Remember, that's our key here. And I think that's it. I'll see you guys in class.